Question number 1. Synthesis of DNA is also known as duplication, replication, transcription, translation. Correct answer is option B, replication. Synthesis of DNA is also known as replication. Question 2. Replication of DNA is conservative, semi-conservative, non-conservative, none of this. Correct answer is option B, replication of DNA is a semi-conservative. Question number 3. Direction of DNA synthesis is 5 to 3, 3 to 5, both A and B, none of this. Correct answer is option A. Direction of DNA synthesis is 5 to 3. Question number 4. Formation of RNA primer. Precede replication, follow replication, precede transcription, follow transcription. Correct answer is option A, precede replication. Question 5. Okazaki pieces are made up of RNA, DNA, RNA and DNA, RNA and protein. Correct answer is option C. Okazaki fragment made up of RNA and DNA. Question 6. Okazaki pieces are formed during the synthesis of mRNA, tRNA, rRNA, DNA. Correct answer is option D, DNA. Question 7. After formation of replication fork, both the new strand are synthesized discontinuous. One strand is synthesized continuously and other discontinuously. Both the new strand are synthesized continuously. RNA primers is required only for synthesis of one new strand. Correct answer is option B. One strand is synthesized continuously and other is discontinuously. Question number 8. An Okazaki fragment contains about 10 nucleotides, 100 nucleotides, 1000 nucleotides, 10,000 nucleotides. Correct answer is option C, 1000 nucleotides. Question number 9. RNA primer is formed by the enzyme ribonuclease primase, DNA polymerase 1, DNA polymerase 3. Correct answer is option B, primase. RNA primer formed by enzyme primase. Question number 10. In RNA, the complementary base of adenine is cytosine, guanine, thymine, uracil. Correct answer is option D. Uracil is the complementary base of adenine in RNA. Question number 11. During replication, the template DNA is unwound at one of the end, at both the end, at multiple sites. Now where? Correct answer is option C, at multiple sites. Question number 12. During replication, unwinding of double helix is initiated by DNAA protein, DNA-B protein, DNA-C protein, Rep protein. Correct answer is option B, DNA-B protein. 30. For unwinding of double helical DNA, energy is provided by ATP, energy is provided by GTP, energy can be provided by either ATP or GTP, no energy is required. Correct answer is option A, energy is provided by ATP. 40. Helicase and DNA-B protein cause rewinding of DNA and require ATP as a source of energy. Rewinding of DNA but do not require any source of energy. Unwinding of DNA and require ATP as a source of energy. Unwinding of DNA but do not require any source of energy. Correct answer is option C. Unwinding of DNA and require ATP as a source of energy. 50. The unbound strands of DNA are held apart by single strand binding protein, double strand binding protein, Rep protein, DNAA protein. Correct answer is option A, single strand binding protein. 60. Deoxyribonucleotides are added to RNA primer by DNA polymerase 1, DNA polymerase 2, DNA polymerase 3 holoenzyme, all of these. Correct answer is option C, DNA polymerase 3 holoenzyme. 70. Ribonucleotides of RNA primer are replaced by deoxyribonucleotides by the enzyme DNA polymerase 1, DNA polymerase 2, DNA polymerase 3 holoenzyme, all of these. Correct answer is option A, DNA polymerase 1. 80. DNA fragments are sealed by DNA polymerase II, DNA ligase, DNA gyrase, DNA topoisomerase II. Correct answer is option B, DNA ligase. 90. Negative supercoils are introduced in DNA by helicase, DNA ligase, DNA gyrase, DNA polymerase III holoenzyme. Correct answer is option C, DNA gyrase. 20. Reverse transcriptase activity is present in the eukaryotic DNA polymerase alpha, DNA polymerase gamma, telomerase. Correct answer is option C, telomerase. 21. DNA polymerase 3 holoenzyme process polymerase activity 3 to 5 exonuclease activity 5 to 3 exonuclease and polymerase activity 3 to 5 exonuclease and polymerase activity. Correct answer is option D, 3 to 5 exonuclease and polymerase activity. 22. DNA polymerase 1 process polymerase activity 3 to 5 exonuclease activity 5 to 3 exonuclease activity, all of these. Correct answer is option D, all of these. 23. 3 to 5 exonuclease activity of DNA polymerase 1 is remove ribonucleotides, adds deoxyribonucleotides. Correct error in replication, hydrolyze DNA into mononucleotides. Correct answer is option C, correct errors in replication. 24. All of the following statements about RNA-dependent DNA polymerase are true except it synthesizes DNA using RNA as a template. It is also known as reverse transcriptase. It synthesizes DNA in 5 to 3 days direction. It is present in all the virus. Correct answer is option D. It is present all of the virus. Is not the true statement about RNA-dependent DNA polymerase. 25. Reverse transcriptase catalyze synthesis of RNA, breakdown of RNA, synthesis of DNA, breakdown of DNA. Correct answer is option C, synthesis of DNA. 26. DNA A protein can bind only to positively supercoiled DNA, negatively supercoiled DNA, both A and B, none of these. Correct answer is option B, negatively supercoiled DNA. 27. DNA topoisomerase 1 of E. coli catalyze relaxation of negatively supercoiled DNA, relaxation of positively supercoiled DNA, conversion of negatively supercoiled DNA into positively supercoiled DNA, conversion of double helix into supercoiled DNA. Correct answer is option A, relaxation of negative supercoiled DNA. 28. In mammalian cell cycle, synthesis of DNA occurs during S phase, G1 phase, G2 phase, mitotic phase. Correct answer is option A, S phase. 29. Melting temperature of DNA is the temperature at which solid DNA becomes liquid, liquid DNA evaporates, DNA changes from double helix into supercoiled DNA, native double helical DNA is denatured. Correct answer is option D. Native double helical DNA is denatured. 30. Melting temperature of DNA is increased by its A and T content, G and C content, sugar content, phosphate content. Correct answer is option B, G and C content